So, as the numbers say for themselves there, not too hot. So, a couple weeks, we're going to have a midterm exam. What kind of questions you reckon are going to be on that? Huh? Not a rhetorical question. You tell me, what kind of questions do you think will be on that test? What kind of questions did you think would be on a quiz? What kind of things did I tell you I would ask you on a quiz? N, little n, big N, alleles, genes, loci, do a ratio. Knuckle down and get this stuff learned, okay? Or it's not going to be pretty. Portia? All right. Shots downrange or downrange. So make your next shot better. So we're going to finish talking about weird things that happen to chromosomes. One weird thing that can happen is when two species that are related enough that they could interbreed, but they are different species. So biological definition of species, as it's stated, would be two groups of animals that could not productively interbreed. Okay, they might be able to actually have offspring, but their offspring are not able to reproduce. And that would be one way of defining a species or defining a pair of species. So with, say, like horses and mules, you have different numbers of chromosomes, or you have a couple of different salamanders that can interbreed, but they have different number of chromosomes, or something like that. Well, what happens is, since each of the parent species produce a haploid gamete, if those gametes are compatible enough that they can biologically fuse and their mating rituals are good and close enough or whatever that they can actually interact, well, then you're going to be able to produce an offspring that has one of every one of the chromosomes from parent one and one of all the chromosomes from parent two, but the two numbers of chromosomes that come from each parent aren't the same. Okay, so this is known as an alloploid, and it has all the right number of genes, but those genes are not arranged on homologous chromosome pairs. So none of these are pairs. These look similar, but they don't necessarily contain exactly the same sets of genes. But taken collectively, all of the necessary genes are there. Okay, so horses and mules can be an example of this. So here you have a karyotype of a horse, karyotype of a donkey. So these are two related species, Equinus calibus and Equus asinus, and they have different numbers. They have different N numbers. Okay, there's 32 different types of chromosomes in a horse. There's 31 different types of chromosomes in a donkey. If I don't know where I got this picture, but clearly a male horse and a male donkey are not what are getting together here. Well, one would have to be a female, but in any case, the offspring is going to have one set of horse chromosomes and one set of donkey chromosomes. Okay, if they get an XX, then they're a female donkey. It's called Jen, Jen, or Jenny, Jinni, doesn't matter, or it's going to be a male, which is just a mule, or a jack. And in this case, you can see how there are chromosomes that would look similar, but they're not actually homologous. They won't pair up during meiosis. And because of that, meiosis is just not going to happen. You're not going to produce viable eggs or sperm, and therefore the mule is a sterile animal. It doesn't produce any gametes, okay? Or if it produces gametes, the chance that they are going to be useful would only happen if they got either all the donkey chromosomes happen to segregate into a gamete, or all the horse chromosomes happen to segregate. I mean, what are the chances that they go just perfectly and segregate like that? Very small, okay, because it's a random event. So you basically have a half times 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 a half. 63 halves, okay? Very unlikely that that's going to occur. And so for all intents and purposes, mules are sterile animals. And that's often the true of any sort of hybrid like this, which is the result of an interspecies mating where you create an allopoid offspring. Is it possible for two recognized species, different species, to have progeny that are fertile, that are going to be able to reproduce? And the answer is yes, okay? And this is generally where two species have not got different number of chromosomes, but are recognized still as two species. When they can reproduce and the offspring are fertile, you have to go back and rethink. Maybe they aren't really two species, and that's one reason why we might change them from being two different species to being, say, subspecies, because subspecies are able to interbreed. So a couple of different types of species of antelopes, I think these are. And they're different species because they live in geologically distinct regions, and they never actually have any normal opportunity to interbreed, right? One lives way far away from the other across mountain ranges. They're not going to get together on a day-to-day basis, but this was found out after they were put in zoos and things like that, oh, hey, they're perfectly happy to get together. And you can see what the allopoid offspring looks like. Its karyotype shows that you have pairs of homologous chromosomes. They have exactly the same number. They both have 30 chromosomes. And there's some differences in the sizes and the sort of some of the banding patterns of the chromosomes. But all told, meiosis is going to work fine, and the hybrids, which would be the karyotype here, the hybrids are fertile. Okay, so that would be a case where you'd have alloploidy, but it creates a perfect pairing of homologous chromosomes, so it's known as an allodiploid. Okay, this one's not diploid, not an allodiploid. Okay, because it doesn't have pairings of chromosomes. They're not homologous to each other. All right, so alloploidy can sometimes happen, and it can also come from a non-disjunction event. So instead of each parent contributing a haploid gamete, they actually end up with a diploid gamete. Okay, that's bad news, unless it happens to pair up with another diploid gamete and they can somehow survive. This has happened a few times, we surmise, based on the chromosomal composition of a bunch of different animals that we've studied. In plants, this happens a lot in plants. But the frogs that we use in developmental biology are called pseudotetraploid. They resulted from this type of an event. they essentially have two number ones, two number twos, two number threes, two number fours, but all of the genes, let's say, that's present here are also present here. So they are, in fact, tetraploid. And they're known as an allopolyploid. Sometimes they're also known as a pseudo-tetraploid because they do not have exactly 4 of the exact same chromosomes, but they have 4 sets of genes. Okay? So that would have to have, this animal would have had to have produced a diploid gamete for this to occur. And that's generally not going to happen. But in animals that make bazillions and bazillions of gametes, like fish and frogs, and lots and lots and lots of pollen and ovules like plants do, it can sometimes happen and has, and creates what are known as allopolyploid organisms. So an allopolyploid organism has 2 complete sets of genomes. It has the complete genome of each parent, or it has the complete set of chromosomes, I should say, from each parent. Okay? Instead of having 1 from each parent, it has 2 from each parent. Okay, so those are all differences in chromosome number. Okay, what is the name given to any alteration in the normal number of chromosomes? Say it louder. I can hear you. Well, you said something. Now say it again. Just say it louder. What? Say it louder. Louder. Oh, come on, yell. I'm going to be here all freaking day. Aneuploidy. There you go. Use your outside voice. Now, why are people so timid? Nothing to be scared of. All right. Aneuploids. Now, that's a wrong number, but what if chromosomes just get jacked up and there's a piece missing? That's a deficiency. There's extra region, that's duplication. Okay? In those 2 instances, the amount of material is changed. You've either got extra or you lost it, right? There's 2 other ways. You can have an inversion or a translocation where chromosomes get rearranged, but you don't typically lose any genes. You just move them, so they're still there. So the genetic material's there. It doesn't mean it's going to necessarily work perfectly because moving it could make it not work, but it's still there anyway. You haven't lost anything or gained anything. So deficiencies, duplications go together, inversions, translocations go together. This constitutes 4 chromosomal aberration types that we'll talk about. Okay, so when a chromosome breaks, like if you just it somehow got broke down here on the end and you lose that little bit, you're just going to have a shorter chromosome. That's called a terminal deficiency because it broke off the end, the terminus. It could break in the middle, okay, but it would have to break in 2 places. So if it broke here and broke here, but then the tip got rejoined, you would have lost some sequence in the middle. This is a more rare instance because it requires 3 events. It requires breaking, and then it also requires rejoining. And it's the rejoining part that's the problem. But if that happens, you would get what's called an interstitial deficiency. Interstitial means in the middle, in between. So in either case, you're going to lose genes. Probably lots of genes, or at least several genes, and which would be probably noticeable in the phenotype if you had a defect on the other chromosome as well. If this happens only on one chromosome, most gene losses are recessive, so you'd probably be okay. Here's an example of an instance when it's not okay. Okay, so there's just a little bit on the end of chromosome 5 that is lost. and it results in a phenotype known as Creuset. And the deletion is not always exactly the same size. So it could be a little bit more or a little bit less, and the symptoms go along with that. More deletion, more detrimental syndrome symptoms. So in a person with that loss, that's the way you would write that karyotype. You count the total number of chromosomes, put the sex chromosomes. This could be male or female. This just happens to be female. And then you put that 5P has a missing piece. So 5P, that tells you chromosome 5, the short arm, and then it's missing a piece. It doesn't tell you exactly what part's gone, but it tells you that it's gone from the end. Okay, that's one example. And even if the person is heterozygous like this, they will have the syndrome symptoms. How can you generate these losses? Or what kind of things cause chromosomes to break like this and for pieces to be lost? Let's say most chromosome damage like this is caused by ionizing radiation. So radiation is ionizing radiation, right? Chernobyl kind of stuff. And cosmic rays, things that are higher energy, not UV radiation, that damages DNA, but it doesn't damage it by causing breaks. It causes different kinds of damage. But to break molecules of DNA requires pretty high energy radiation. Okay, but it can happen during meiotic recombination. These are going to be smaller scale alterations, but it can happen. And that's going to be something known as unequal crossing over. You have chromosomes that have a set of genes on them, okay? So here are two homologous chromosomes, but there are regions that are shared or that are repeated. So if there were repeated sequences located between B and C, also located between C and D, then those two sequences could line up slightly off, okay? So if this region between B and C on this homolog got lined up with the region between C and D, and then recombination happened to occur, you would end up with unequal recombination or unequal crossing over. So this chromosome would break here after C and rejoin to this chromosome before C. And you would end up with two Cs. You would have doubled that gene, plus everything else in that little region. Okay? And on the other chromosome, you would have lost the C. Okay, so you would go from here, you would go B, and then you would jump up and then pass C and go to the D. So you would end up with a chromosome with a deletion. Because this is only happening between one pair of non-sister chromatids, you also get the sister chromatid that has no recombinations, and those are fine. But two of the chromosomes are going to be abnormal. One would have a duplication, and the other would simultaneously have a deletion. And that's how a lot of duplications and deletions have occurred. And we can track it because you can see right where the chromosomes broke, and you can see what sequences were there. And what you see is that the same sequence is present at two different places on the chromosome. These are sequences that aren't genes. They're known as repetitive elements. Repetitive elements. And so they're repeated, right? That's what that means. And they're not necessarily small. Some of them can be thousands of base pairs long. And if they're pretty long, then the chances of them accidentally getting lined up incorrectly go up. The longer the sequence is, the more likely it might get misaligned. So the good news is, most of the time, these sequences just misalign with themselves and they're much more prevalent than the genes. So you end up with recombinations where the repetitive DNA varies, but you don't lose any genes or gain any genes. And this happens all the time. And in fact, it's immensely useful. It's what we use. The variations like this are what's used in a lot of DNA fingerprinting. So you look for these very small changes which would happen between individuals, and there's lots of places for them to change. So the chances that any two individuals who aren't twins or offspring, parents to offspring, have exactly the same combination is real low. It's like 1 in 7 or 8 billion. We're getting up there now where there's that many people on the planet, but right now it's still pretty good. So unequal crossing over can cause a deletion and a duplication. They're generally going to be relatively small scale, maybe only a few genes, maybe just one gene, maybe no gene, but just DNA. Okay, that's not a gene. So again, the big thing about deletions is that chromosome breaking has to happen. Normally it happens during meiosis, but if it happens in other cells outside of meiosis, there's probably an outside cause, like ionizing radiation, X-rays, gamma rays, cosmic rays, that kind of stuff. Now, what are some consequences of these alterations? Duplications and deletions will have potential phenotypic consequences. We saw the Cretice phenotype. Duplications tend to be less problematic than deletions, right? Having an extra gene is not probably as bad as not having the gene at all. And however, if you have too much of the whole chromosome, we saw, we talked about that Tuesday, right? That's very bad, right? There's only 3 autosomes that are even survivable for humans if you have an extra. But you're not talking about 1 or 2 extra genes. You're talking about hundreds of thousands of extra genes. And so it just jacks up everything. You get a couple of extra albumin genes, you're going to be fine. Good as rain, okay? But if you get a whole extra chromosome, that could be bad news. So duplications are a little less problematic than deletions. Here's an example of a duplication that has a definite phenotype for fruit flies anyway, and it's a region of their X chromosome where it's supposed to be inactive. Okay, so they're showing that by this little ball area there. It's supposed to be inactive, and you're supposed to have 2 X chromosomes if you're a female, or 1 if you're a male, and that region's supposed to be inactive, and that's all good. And if that's true, the genes next to this are part of them are involved in making the eye have the normal number of little ommatidia, little actual working functional units, right, in a compound eye like a fruit fly has. So that's great. If you're normal, you have about 780 of these little facets in your eye. Well, if this region gets duplicated, which can happen very easily by unequal recombination. So these could line up in a way where you actually get recombination, you get a chromosome that has 2 of them. Well, now the fly has 3 areas that are supposed to be turned on. And this starts to turn off genes that aren't supposed to be turned on. It sort of expands its reach. And you end up with smaller than normal eyes. If you have 4 copies, you have even smaller eyes. And it doesn't matter if the 4 copies are on the same chromosome or on 2 separate chromosomes, you still end up with really small eyes. This is known as bar, okay? And it was called a gene, but it turns out that it is not even a gene. There is no gene here, but there are genes next to it. And this area, as it's being compacted, can accidentally draw in the area next to it and compact it improperly. So it's like losing those genes because they're not being active. That's an example of a duplication. The cool thing about duplication is they really have been important in the evolution of vertebrates. We're vertebrates, so you should perk up and go, oh, That's us. In making extra sets of genes to do cool things, okay, extra things. So if you had one gene for a thing or two copies for a thing and it got mutated, you can't do that thing, right? But if you had duplicated that gene and one of them mutated and the other one didn't, then you still can do the thing, plus you could do whatever the mutation might be able to do. You could do something extra, right? It's like if you have $1, all you can do is, you know, get half a candy bar out of the candy machine. If you had $2, you could get half a candy bar out of the candy machine and half of something else out of the candy machine, right? You could expect. It's a dumb analogy. But if you have two genes, two copies, three copies, evolutionarily, if they mutate, they could start taking on different functions. Or they could take on different functions without a loss of the original function, right? So this creates gene families. And so you have, anytime you have two or more genes that are clearly share homologous sequences or similar sequences, they may have arisen by duplication. And we can track them by sequencing DNA. So we say that they were probably derived from a common ancestral gene that at some point was duplicated in an animal's genome somewhere along the way. And we call them homologous genes because homologous means from a common origin. So you have a gene and maybe there's just one of those little unequal recombination events and you get two copies of that gene. Now they start to mutate. But because they're not going to mutate identically, you're going to have changes in two versions, and each version may be able to be subtly function different. The protein that it encodes may function subtly different. Or the genes might not change the protein at all. They just change where the protein is made or when the protein is made or how much of the protein is made. Okay? So a lot of times we think about when a gene is mutated, it means the protein has changed. That's true, but not always. Sometimes it just means the gene is turned on differently, turned on more, turned on in a different place in the organism. Okay? Think about all those different variations. And a very subtle change in the DNA could really have a dramatic change on how much of it is made or where it's made. And that will have a much bigger impact on the development of the organism and the evolution of new body plans than changing the protein, okay? So what are some gene families? We should know them, right? There's the Sackville Bagginses. No Lord of the Rings fans in here? You've heard of Lord of the Rings? The Hobbit? Okay. Stay with me. All right, so you've got the globin gene family. Right? Just move that L around and what you got? Goblins, right? Lord, see, I wasn't that far off, was I? So globin, that's the proteins that make your hemoglobin and myoglobin, and there's a bunch of them. There are 14 different genes in humans that were probably derived throughout evolution by duplications and then duplications of the duplications, things like that. So they all bind oxygen. You know what hemoglobin does. So here they are. On humans, we have 3 clusters. There's one gene on chromosome 22, that's myoglobin, and it's made exclusively in our muscle cells to hang on to oxygen in the muscle cells, especially skeletal muscle and cardiac muscle. And then you have the alpha globin cluster and the beta globin cluster. And these two probably had a cluster, and then the cluster was duplicated by adding an entirely extra chromosome, okay? Because we see lots of other families are on these 2 chromosomes. Some of the genes are still active. The orangish ones and the darker blue ones. The gray ones are no longer functional genes. the protein could still be encoded, but the gene is never activated. So it's known as a pseudo gene. Pseudo gene, that means fake gene, right? Well, it's not really a fake gene, it's just an inactive gene. And so it doesn't code a protein anymore. So that's represented with a little Greek, whatever that letter is, psi, I guess. The other genes are functional, but they work at different times during embryonic development. So think about it. In mammals, you're in the uterus, and you got to connect to the placenta, and you got to absorb oxygen from another bloodstream, not by breathing it into your lungs through the air. So you need different functionality. You need the ability to bind oxygen in different ways, and that's what these genes do. So the very first genes to be expressed as the circulatory system is developing are the, whatever that is, and epsilon. And then after, in the second and third trimesters of pregnancy, of development, the fetal stages, the alpha 1 and the gamma betas come on. And then once, just before you're born, and then after you're born, you have your regular alpha 1, alpha, which is alpha 2, the alpha gene we have right now, you have right now, and the beta globin gene that's active right now, coming together to make hemoglobin, okay? So it's thought that a long time ago there was some sort of ancestral globin gene, and that there were duplications that might have set the pathway off towards myoglobins in one way and hemoglobins on one way. There's actually something called phytohemoglobin, which plants have, okay? And they use it to bind oxygen, right? Because plants, you always say, oh, plants produce oxygen. They do, but they need it also. They actually also use oxygen. And so they have oxygen binding proteins also. They don't have blood, but they do bind oxygen in their cells. And so then the hemoglobin side of things duplicated again. And then within those, you had micro duplications, and you have the whole, all this grouping, okay? And this is pretty useful because if you look and compare humans to other primates, to other great apes, and then other more distantly related primates, and then to something like a pig or a whale or a sheep or a cow or some other mammal, you can see there's been changes in which of these get used, okay? And there's sequences in between the genes that can be compared. These two groups have been used to really say we're a little bit more closely related to chimpanzees than we are to gorillas, okay, in terms of the other great apes. And it's because of some common sequences we have in our globin clusters that chimpanzees still have, but gorillas do not have. So that means, well, either we both changed the same accidentally, or we still have something and they lost it, or we gained something both and they didn't have it, or whatever. But it means we have something in common, so that makes us more closely related. I think that, oh, that's right here. So here we are, where are we? Are humans on, oh, there we are, humans. Yeah, well, we're not that related to other things, but if you look here, so hedgehogs and shrews, there's a difference. Cows and pigs and dolphins and killer whales and so forth. You can kind of see some things. So the alpha cluster and the beta globin cluster have changed around a little bit. Here's another gene family, maybe an even more important gene family, okay? If that gene family screwed up, you can't breathe, but if this gene family screwed up, you don't need to breathe because you don't have a head. So in fruit flies, there was a group of genes that were initially identified because they messed up the identity of structures like the mouth appendages, or the legs, or the wings, or the little things behind the wings called posteriors, or the segments. They didn't form right. And so they were called homeotic mutations. Homeotic mutation. And it was then when the genes were mapped, it was noticed that, oh, they actually all map on the same chromosome, kind of nearby each other. And so here's a bunch of them. And then it was many years later discovered that they had related sequences to genes in mice and humans and frogs and chickens. And it was noted that in mammals that we had more genes that were related, and we actually have 4 clusters instead of 1 cluster. So we have A cluster, B cluster, C cluster, and the D cluster on those 4 different chromosomes in humans. And they're exactly conserved in mice and in shrews and in hedgehogs and any other mammal. They have 4 clusters. The chromosome number will be different. 4 clusters. Within each cluster, there are a variety of genes which are all similar but distinct. Okay? And then they're labeled A or B or C or D based on the cluster they're in. And then 1, 2, 3, 4, 5, 6 based in the order they're in. These genes are all located immediately or nearly immediately adjacent to one another on that chromosome in that order. Okay, so in fruit flies, you have this gene, then a little bit, skip a bit, and you have that gene, so on and so forth. The cool thing is, the gene that's at this end of the chromosome is used to direct the development of the most anterior structure, which is the mouth parts. The next gene is used to develop for the development of the next part of the head, and the next part of the head, and the next part of the head, and then the first leg, then the second leg, then the third leg, then the abdomen, first half, and then the second half. So the order that they are in is the same exact order that they control in the body. And that was like, coincidence. Until they found them in mice, and lo and behold, the first gene controls the development of the top part of the vertebrae in the neck, and the next gene controls further down, and the next gene controls further down, okay? And then not only do they control the anterior to posterior developmental patterns, they control the proximal distal developmental patterns. And so these 2 clusters, you can see that basically 8 through 13 are present. Every gene is there. But 1 through 7, we've lost a few. And if you look at these clusters, 1 through 7 are all there, but 8 through, we've lost a few, okay? So as it turns out, these control mostly anterior posterior development, and these control mostly proximal distal development, okay? So if you lose one of these genes here, for example, you'll be missing a piece of your arm. This part will be shorter. If you lose some of these, you get messed up fingers. If you mutate one of these, you get extra fingers. Polydactyly is caused by D13 mutation, okay? If you mutate some of these, you get defects in the body structure. For example, in mice, if you take out some of these genes, you get ribs forming on vertebrae that aren't supposed to have ribs, right? We got 7 cervical, and then 12 thoracic, and 5 lumbar, and you get ribs coming out of the neck vertebrae. I mean, they're not full-fledged ribs, but they're little protrusions that aren't supposed to be there, and you get ribs forming all the way down. Now, pretend you're a snake. How many vertebrae on a snake do they have? Is it constant or is it variable? Variable. And do they have ribs on all of them? All of the thoracic vertebrae do have ribs. They don't have any lumbar ones. They have tail ones, but everything has a rib. You get a mouse that basically looks like a snake. It's got vertebrae on every rib. Okay? Pretty cool. So mutating these genes, losing them, mutating them, can cause abnormalities along the anterior posterior developmental program, or the proximal distal, like in the limb appendages in the program there. Well, it turns out that these genes are almost a 1-to-1 correlation in their order, and in their functionality to the fruit fly version of those genes. And if we talk about these, we have some new terms to describe how genes can be similar to each other between different species, and then multiple copies within the same species can be described, okay? So every gene has its name, but if you want to talk about how they're related, we need these terms. So within a cluster, they're homologs or homologous. If you look between species, so if you took the labial gene, LAB, that stands for labial, which is mouth, lip, and compared it to A1 or B1 or D1, it would be more closely related to A1 than A1 is to A2. So here's a gene from a fruit fly and a gene from a human that share more sequence than two genes from a human that had the same ancestor. Why would they still have so much sequence shared? Why would they need to be the same? Why do all hammers have a head on one end and a handle on the other end? All hammers are more alike than a hammer is to a wrench, aren't they? Why are they alike? They have the same function. So if two genes are very similar in sequence and they encode proteins that haven't changed over the amount of evolutionary distance between insects and mammals, that's an important thing to note. It means that those proteins must do a very similar job. If they change, the organism didn't exist. Therefore, it didn't get passed on. So the fact that only organisms are alive that have that function, the little protein, means that function is necessary. Okay? So we need a name for that. We call that ortholog. A gene in, say, humans compared to its functional equivalent in a fruit fly or a mouse, that would be called an ortholog. Now, in cases where we have multiple families, multiple clusters like this gene, we use a term to describe the members which are most similar within the clusters, between the clusters. So A1, B1, and D1 are all more similar to each other than any of them are to their adjacent neighbor. So A1 and B1 and D1 are almost identical. I mean, they're like 99% the exact same amino acid sequence, okay? But comparing A1 to A2, you're talking like 85% identical. So pretty big difference. So we have the word paralog. Paralog. So 1, 1, 1 are paralogs. 2, 2 are paralogs. 3, 3, 3, 3, what the hell? 3, 3 are paralogs. 4, 4, 4, 4. You notice there aren't very many that have all four. 4s and 9s and 13 are the only ones that have all four groups have all four genes. So why would you put the 2 here? Why would it should be there. They jacked it up. Or actually it is a 3. There isn't a D2, it's a D3. They jacked the number up. Why would you call it D3 instead of D2? Because you compare it up here and it's more similar to the other 3s, right? So that's how this arrangement is made. It's kind of cool. There's 39 total genes. And every one of these genes has been removed individually from a mouse genome. And you look at the consequence of that removal. Removing a gene from a genome is called a knockout. It's kind of cool, knockout. You knock out that gene, okay? And you look at the phenotype. Interesting ones, like I said, can cause ribs to grow in the wrong places. They can cause the pelvic girdle or the pectoral girdle not to form. Like you don't make shoulder blades, you don't make a pelvis. hip bones, things like that. They can cause dramatic alterations. In fruit flies, they cause things like the antenna to turn into legs. Right? You got these little legs sticking out of your head. Like, you think, well, maybe that's good. You know, if they walk on their heads, that'd be fine, but they're not little circus stars. So you can cause, I don't have pictures, you can cause 4 wings to form instead of just 2 wings. Right? Butterflies have 4 wings. Dragonflies have 4 wings, but flies have 2 wings. That's why they're called flies. So you can cause a lot of weird things happen when these genes get mutated. All right, so the Hox gene cluster and the terms homologous. Homologous means from a common ancestor, common origin, right? So these are just any genes that are similar. So 1 and 2 and 3 and 4 are all homologous. Okay. 1 is also homologous to labial because they probably shared a common ancestor. But we don't use the term homologous because that doesn't tell you if you're comparing 2 species or the same species. So that's why we threw in the term orthologous. So orthologous genes are homologous by definition. But orthologous just tells you, yes, they're homologous, but they're in 2 different organisms. Pretty different organisms. Does that make sense? It's just an extra way of describing it. And then paralogous is just because there's 4 groups, right? So fruit flies don't have anything about paralogous because they just have the 1 of every gene. They don't have 4 labial genes, right? But mice, mammals are the only thing where that matters. Mammals have 4 copies, okay? I guess actually vertebrates have 4 copies. Chickens do and frogs do too. All right, so that's duplication. Pretty cool, huh? You make families. They can go on game shows, have feuds. Great. All right, so what about other rearrangements? You can have inversions. An inversion would require 2 chromosomal breaks. What happens if the chromosome breaks twice and the piece of DNA is lost? What do you have? A deletion, an interstitial deletion, right? So inversion and interstitial deletion are kind of related. You probably had the same reason the chromosome broke, but with the inversion, it got reconnected before it got lost. And it may have just got reconnected backwards, okay? And so it's inverted relative to the homolog. The sequence of genes are still there. They're just in a different order now, okay? And so that's known as an inversion heterozygote, right? You're heterozygous, but what you're heterozygous for is not necessarily the gene version, but just the gene position, okay? And usually there's no phenotypic consequence. In fact, it's estimated that about 2% of the human population would have a detectable inversion. Very likely 100% of the population has inversion, but it's just not detectable. It's so small. Now, what happens if an individual has an inversion? They won't have any problems phenotypically, but they could run into problems when meiosis happens because the chromosomes might not segregate properly if recombination occurs. If recombination occurs in this inverted region, it can wreak havoc on the chromosomes that are formed, and essentially you make half the number of viable chromosomes, and that means half the number of viable gametes. So it means a small reduction or 50% reduction in, say, fertility. And so a lot of times these are actually diagnosed when couples maybe have been trying to have a baby and just haven't been having much success. And so they get checked out and everything perfectly checks out with their reproductive tract. There's no blockages, there's no tight genes, it's all good to work functionally. And they go, well, now we maybe need to go a little deeper, and they ask about, let's do a karyotype, and it turns out that one or both have an inversion. And all that means is like everything works, it's just you're just not making very many gametes. And so the chances go down, right? So just keep trying, try more often. Oh, yeah, so the centromere is inside the piece that flipped around here, and it's not. So that's the only difference. Para is off to the side, peri is inside. Are these homologous? Yes, they would still be considered homologous. Yep. So what would happen if there was a gene right where, like here's a gene and it broke that gene, right? Now that would cause a problem. That one gene got messed up. That's called a breakpoint effect, right? Another problem would be you don't break a gene, but let's say this side is inactive area, and you took an active gene and you plopped it into an inactive area. That gene might be there, but it won't get turned on. So it's kind of like you deleted the gene. So that's called a position effect. You don't delete the gene, you just move it to a place where it don't work anymore, okay? So what could go wrong with inversion? Breakpoint effect, deletion of position effect, and problems with recombination. We'll look at that here. Okay, so here's a couple of chromosomes. One has an inversion here. The BCB is flipped, okay? Gotta look careful and see that. All right. If they line up to synapse, the B is going to try to go with the D is going to try to go with the D. So one of those chromosomes is going to basically kind of contort itself, redraw it. This is a schematic. This is not how the chromosome actually looks, because you cannot stretch. You cannot stretch the chromosome. So both chromosomes are actually going to be contorted, but we just draw one of them being contorted to create a loop. Okay, so this is known as this is just a diagram. It's not what really it looks like. So now we get to line up the B's, the C's, and the D's on the replicated chromosomes. So you've got the sisters from each homologous chromosome paired up here, right? So what happens if recombination occurs somewhere here, like between B and C, which it could? Well, now let's track the crossover. So if we say, let's number each end of the chromosome, 1 and 1 prime, so that's the same sister chromatid. Each sister chromatid gets the same number, just like nothing in a prime on each end. Okay, follow me? So now it's track number 1. It goes down, has a centromere, A, B, C, D, E, F, G, H, I, boom. So number 1 is going to be just fine after segregation. Okay? Track chromatid 2, A, B, and then it crosses over, keeps going the same direction, A, B, C, D, A, centromere 4. So you just took chromatid 4 and joined it to chromatid 2 to create a hybrid, right? And that's right there. Okay? Without all the distance drawn in there. That chromatid's really jacked up because it now has 2 centromeres and it's missing a whole bunch of genes. Okay? Track number 3, 3 comes in, loop-de-loop, right back out to 3 prime. 3 has no changes on it. So draw it down here, 3. Regular original order that it had up here. Okay? It is inverted, but it's not rearranged. All right, now look at what's left. When this recombination occurs here, you're going to join the other end of 2 and the other end of 4. So you'll come down here and go, okay, I, H, G, F, E, D, C, B, EFGHI 4 prime. You'll create a chromatid that has no centromere in it. Okay? Now, anaphase, this happens during prophase 1. Now, when anaphase happens, what's going to segregate? This chromosid, this centromere is going to get attached to the chromocentre over here. That one's going to get attached over there. And they're going to get yanked apart, but they're actually connected. So that is going to get broken. It's just going to physically get stretched and broken apart, or it's not going to break, and you're going to end up with disjunction, non-disjunction, because this is not going to get to go. But usually it just breaks, and where it breaks is random. So you're basically going to get just a chunk of chromatid that's no good. Okay, 2 broken chunks. Those are going to get degraded. They don't have a telomere on this end. Broken, damaged DNA is generally just degraded in the cell, especially if the nucleus is gone, which is gone at anaphase, right? So that gets degraded, lost. What about this one? It'll end up in a cell, but who knows which cell it'll end up in. There is no centromere. It might not ever end up back in a nucleus. In fact, it probably won't. So if it's just floating around out in the cytoplasm, it's probably just going to get degraded. So that entire chunk is lost. So what are you left with? The only things that could possibly end up in a nucleus of a daughter cell during meiosis are the original chromatid that you had. You had the regular alphabetical order and the inverted order. You won't get any that are rearranged. And all the ones that are rearranged are either going to get destroyed or lost. And so you're only going to be able to make a gamete out of one of those 4 possibilities, which means, or 2 out of those 4 possibilities, which means statistically half the number of viable gametes. And none of those viable gametes are going to have any rearranged genes in that area. Now they can cross over out here, no big deal. So there'll be some rearrangements, but they won't have any rearrangements in that area. We say that the inversion suppresses recombinant phenotypes. Okay, it represses the formation of those chromosomes, which prevents the genotype, the recombinant genotype, which of course then prevents any recombinant phenotypes. This is the paracentric inversion. The paracentric inversion is the most deleterious because it really messes up all the products except the original chromosome arrangement. Okay. If the rearrangement is in the pericentric, where the centromere is in there, it still jacks up everything, but you do get 4 chromatids that have centromeres on them. You can undergo normal segregation, but the products that are produced at the end of segregation will have chromosomes that are missing chunks of DNA or chromosomes that have duplications. This is another way that you could get, theoretically, a duplicated area of a chromosome. The deleted areas are probably going to not make a viable gamete, but the duplicated areas probably would. So this one might make it, that one probably won't. But in any case, you only get 2 normal, you only get 2 that have every gene in an arrangement, and that's the original parental arrangements. Okay? So again, the likelihood of getting a recombinant genotype and then the recombinant phenotype go down, right? What do we use crossing over to do? What do we make use of crossing over for in genetics? It has its biological function, which is genetic variability. What do we use that for in genetics? That we talked about in chapter 5. Mapping, okay? You can't map with an inversion heterozygote. You will not get recombinant chromosomes, so you won't get any recombinant phenotype. It makes the chromosomes look like there's complete linkage between the genes in that area. You can use that on purpose to keep mutant genes adjacent to one another. So if you have a mutant C and a mutant B, and you always want to keep mutant C and mutant B next to each other on a chromosome, you could take that chromosome and pair it with a recombinant or inverted chromosome, and you'll never get a rearrangement of those genes, because if you did, the offspring would die. Okay? So you only get the original arrangements. Here's what it might look like in real life. No loop-de-loop. The chromosomes are bent and contorted. So you have these homologs and these homologs trying to line up, okay? So only the parental chromatids or chromosomes will survive the whole process, right? So you get no recombinant phenotypes when inversion heterozygotes are mated. Pair-eccentric, you get duplicated, deleted. Pair-acentric, you get acentric, means no centromere, and dicentric, meaning 2 centromeres. This is super not good. Okay, they never will survive that. All right, so the other rearrangement is translocations. Okay, how do chromosomes deal with translocations? How do they get formed in the first place? So there are 3 different rearrangements that we can note and give them names. One of them is called Robertsonian translocation. A Robertsonian translocation is where 2 chromosomes, almost in their entirety, fuse together. Usually there's a little bit of loss. There is non-reciprocal, also called unbalanced. One chromosome breaks and a piece of it joins to another chromosome, and that's it. Okay? And then the third type is reciprocal, also known as balanced. Okay? So this is reciprocal and non-reciprocal are the normal terms you will hear 90% of the time. Balanced and unbalanced are kind of jargon. 2 non-homologous chromosomes each break and swap places. It's like meiotic recombination happens, but with non-homologous chromosomes. It probably uses the same exact mechanism. Okay, this will happen. This can happen, or be caused to happen, in any cell, anytime. So this could happen due to ionizing radiation in a somatic cell, and then that somatic cell has that rearrangement, and all of its progeny cell will have that rearrangement. Okay. And this can lead to tumors and things like that. Reciprocals happen mostly during meiosis, and Robertsonians, we don't really even have a clue, I think, how those can really ever happen. Okay, a lot of times there isn't going to be a problem because you haven't lost anything, you've just moved its location, except the same problems that you could have with inversion. You could break a gene, so there's a break point effect, or you could move it to a place that's inactive, so a position effect. But like an inversion, this is going to cause a problem with synapses during meiosis. So here's what they can look like. Here's, say, 2 pairs of chromosomes. If a piece of chromosome number 2 joins onto 3, that's unbalanced, non-reciprocal. If number 2 breaks, number 3 breaks, they swap places, swap pieces, that's reciprocal or balanced. Here you have a number 2 and a number 3, and they've just merged, pretty much merged. You lose a little bit, probably no genes there anyway, so who cares? Now you have 3 chromosomes instead of 4, but one of them is a combination. So all the genes are still there, good to go, okay? We are the result of a Robertsonian translocation. So ask yourselves if it's okay. It is. You're here because it is. Didn't save my change. I had a different dude there from the other night, but it didn't save it. Sorry. Every 4 years, this lecture almost always falls right along the lines of a debate. And so somebody's always got the picture of somebody yelling at the other person during the debate. This was 8 years ago, I guess. That was when she and Obama were running together, not when she and Orange Man were running together. So that's an old picture. I had an Orange Man up there. Anyway, we have 23 pairs of chromosomes, and chimpanzees and gorillas and orangutans, the other three great apes, have 24 pairs of chromosomes. And that always interested people, like, for a long time. In fact, up until the mid to late 40s, 1940s, karyotypes weren't that easy to do. And there was a lot of debate. And most people, because you could do a lot of them on primates, most people really believed that all of the great apes, us included, had 24 chromosomes. And it took a long time to really get the precise data to show, no, in fact, we only have 23 pairs. They're like, well, that makes no sense. Why would chimpanzees and gorillas and orangutans have 24? And so which chromosome is different? And so as you map them out, it turns out our number chromosome 1 and an 8 chromosome 1. So we're on the left and the 8 chromosome is on the right. And you can see those two chromosomes are really, really similar in their banding pattern. I mean, identical down to right there. And there's a bit of an inversion that's happened right here, where this part and that part have split, and it just throws the centromere off center a little different between us. And then there's some deletion in this area right here, so everything has been scooted up. I think something got deleted, maybe right there. Everything got scooted up a little bit. But basically, everything is the same. Here's our chromosome 2, and there is no matching chimpanzee or gorilla or orangutan chromosome 2. Okay? If you look at their next largest chromosome, it looks nothing like our number 2, but it looks exactly like our number 3. More exact our 3 than the 1s do. And the 4s look exact. And everything else almost matches perfectly. The only difference is they have 2 chromosomes, and they have been renamed now to be 2A and 2B. Their 2 chromosomes together match up to our chromosome 2. So if you look at their 2A, it matches up almost perfectly with the P arm, and their 2B matches up perfectly with the Q arm. The centromeres would be in different places, and there should be sequences here that we could be able to look at and say, if these 2 chromosomes fused in a Robertsonian translocation, that would have created the human chromosome 2. Are there sequences there? Are there residual sequences that can still be found in the human chromosome and compared to the primate chromosomes, the non-human primate chromosomes, and see if we could actually map where that fusion happened and maybe get an idea about what happened? And the answer is yes. And there's a paper you'll be reading. It's Igido, IJDO, I think, or IDJO, I can't remember. Anyway, in that paper, they explore the sequence of this area of our chromosome, and they find cryptic telomere sequences here that show where most of the telomere was lost, but a residual amount of a telomere was there. And then also down in here, there is a residual chunk of centromere. I think it's in a different paper. But they found both things, and that clearly shows that, yeah, telomere sequence got lost, centromere sequence changed from being a centromere. Otherwise, you'd have had 2 centromeres. We kept this one, that one mutated to not be a centromere, and now we have a chromosome 2 that used to not be our number 3 when we first were becoming not apes, was our number 2, because that would have been the chimpanzee number 2. Okay, so it's kind of cool, isn't it? You're like, okay, was this the Garden of Eden event? Was this, I mean, there's a lot of religious overtones, there's a lot of scientific overtones. It's like, pretty much the same. We yell and scream. Chimpanzees are probably much more peaceful than we are. Well, they're mean bastards, though. Everybody's like, oh, let's have a pet chimpanzee. They kill their owners lots of times. That's great. Go get one. So these sorts of translocations have happened a few times in evolution, and it's kind of interesting. We may be one of them, the great times that it happened. What happens when they try to do synapses? And all hell breaks loose when they try to do synapses. So here you have, let's say, a little example of a 9 and a 14 that have swapped some parts. So here's the regular 9, A through G. Here's the regular 14, R through V. These are just names thrown on there to show. And then these 9 and this 14 have undergone a reciprocal translocation. Okay? So when they try to synapse, these regions are homologous, these regions are homologous, but these regions are not, so they will flip around so that when they line up, they can also be pairing with their non-homologous chromosome that contains the translocated piece. So the blue part of 14 wants to match up with the blue part of 9 that contains the same genes, and the red part with the red part. Okay, so they're actually going to create this little tetrad alignment, and the plane or the meiotic, the metaphase plate could be running vertically or horizontally when that alignment occurs. And depending on the way that is running will dictate how, that's why I wear this, because I want to itch my face so bad. They want to dictate how that moves. Is it going to go this way, top to bottom, or is it going to go this way, left to right? Or in fact, a third way, which is 2 of them are going to go that way, and 2 of them are going to go that way into the board. Okay, so there's 3 possible segregation possibilities there for these chromosomes when they line up. Okay, so the first way we'll look at is sort of out of the board, into the board. So let's say the red lines are coming out towards you, the blue lines are going back into the other cell. This is known as alternate. So you're going to have number 1 come out, number 2 go back, number 3 come out, number 4 go back. Every other one, if you go around in a clockwise fashion, every other one goes into an opposite cell or an alternate cell. They alternate. That will be fine. That will result in offspring that get a complete set of genes every time, no matter what gamete is used. Okay? Got everything? Perfect 9, perfect 14. Perfect 9, perfect 14. Translocation, but you got all the genes. You got A through F, you got T through V. Everybody's good. Everybody's good. Okay? So alternate segregation of a translocation gives you progeny. Okay? That's going to happen roughly 50% of the time. The other 50% of the time, actually, that's going to happen about 40% of the time. They say roughly 50, but I think I've read some other papers where they calculate that's probably about 40% of the time. Don't even really worry about the exact number. Okay, what if you give these 2 and they go, now this time they're going to go up. So red means up, and blue means down. So these are going to go into a cell up there, and the bottom ones are going to go to a cell on the bottom. So the metaphase plate was running right through here. Right? So now what do you get? This is called adjacent 1 because the two that are adjacent are going to go together. So you get a normal 9 and a messed up 14. You're missing some 14 genes. And you have duplication of 9 genes. Here, same thing. Here, you're missing 9 genes. You have duplications of 14 genes. Same here. No gamete is normal. All gametes are defective. This is going to happen about 40% of the time. And then the last 10% of the time, they say very rare, but so 5, 10, maybe 2% of the time, in any case, is alternate or is adjacent, but it's the opposite adjacent. So you have the 1 and 3, the centromeres that are both from number chromosome 9 get pulled to one cell. Let's say we separate left and right. They go left, these go right. You end up with basically no 14 genes, no 14 genes, no 9 genes, no 9 genes. So again, all 4 gametes are jacked. So anything that's adjacent is going to be non-viable gametes. Anything that's alternate is fine. So you get about 40% instead of 100%, right? Viable gametes. So again, it's just a reduction in fertility. No phenotypic consequence generally, okay? All right. You got translocated, duplicated, inverted, deleted, recombined. Why are we here? Robertsonian. You get t-shirts that say, I'm Robertsonian. What are you?
What is
The number of different sets of genes.
oh sure, okay.
Yeah.
And this one is
Epistasis, but it's epistasis. So you had to choose.
so complete dominance is something because
Well,
I was thinking it's still dominant because this ratio comes from 9 to 1. And I was like, okay, it's complete dominant, but it's specific.
but dominance means you have one version. Well, okay. Yeah, I can go with that. I see what you did there. Okay, that's fine. I'll give you points.
Okay. So can I just get one?
I thought you were absent.
I don't have anything. You can just put this over 25.
Over 25.
That or extended deadline for the internet. One of those two requests.
Nice.
Yes. So just have to fix it.
Oh. Nah, you can do it.
One of those two. Okay, so put it over 20.
I'll think about it.
Yes.
Stop thinking.